Okay, so last time we talked a bit about how DNA is replicated at a macro level. But now we're going to delve way deep down to the chemical level and examine the chemistry of what's actually going on. So you see here, right, this is this is a chain of two nucleotides. That's a C and a G. I read them in that order for a particular reason. This is the 5' end. You can tell because the phosphate is connected to the 5' carbon, and it is free. It's not connected to anything else, so that's the 5' end. This is the 3' end, and you can tell that because this 3' OH, it is not connected to anything else. It's free, so that is the 3' end. You usually read DNA from 5' to 3'. There's a reason for that. It's because DNA is made 5' to 3'. That means that whenever you add to a DNA strand, you're going to be adding to the 3' end. The 5' end is going to be facing behind you, and the 3' end is going to be facing in front of you. Now, what is added is actually a nucleotide triphosphate. If it's DNA, then this would be called a DNTP, which stands for deoxyribonucleotide triphosphate. If you're making DNA, that's what you're making it out of. If you were making RNA, it would just be an NTP, but we're not making RNA. So the next base in our sequence that we're going to be adding is thymidine, T. And specifically, we have a DTTP precursor. So this is deoxyribothymidine triphosphate, 3 phosphates. And this is an endergonic reaction. It consumes energy, but the energy that it consumes, the energy necessary to drive this reaction forward, is contained within this triphosphate bond. Many of you are probably aware that ATP is the energy currency of the cell. But in this case, no matter what base it is, because it's actually the triphosphate part of the ATP that contains the energy, no matter what base you're adding, the energy is contained in that triphosphate. What happens is that, for those of you who've had organic chemistry, you have a nucleophilic attack from here onto that phosphate. The electrons get kicked up and around, and that diphosphate, what's called pyrophosphate, is kicked off in a nucleophilic substitution. For those of you who haven't had organic chemistry, just know that this 2-phosphate, double phosphate, really wants to leave, and in order for it to leave, it has to bond to something else, and what's available is this oxygen there. So the free 3' hydroxyl bonds to what's called the alpha phosphate. This is the alpha, beta, and gamma phosphates. And so the alpha phosphate binds in, beta and gamma get kicked out, and it is always the 3' hydroxyl that does the attacking, so that's always the end that you build off of. So, this leads to a problem in in DNA synthesis. So if you'll recall, DNA is built bidirectionally. You start at the origin of replication, and then you build this way, and you build this way. If we take this replication fork and magnify it, you'll see that we need DNA synthesis to proceed like that. We can only build DNA 5' to 3'. Here you see is a 3', and so anti-parallel, the opposite strand that we're building, should be a 5', and we can build in this 5' to 3' direction, no problem. But over here, we have a 5'. That means that this end has to be a 3'. And this end down here needs to be a 5'. If we're building it, we need to build that direction. But we're going this direction. So how do we build this direction while going this direction? And the answer is that you build in small segments. You basically have, so here's our 5', 3'. That's the old strand. I'll do the new strand in red. What you do is you start off here and you build a small segment, and then you jump off and go back here and you build a small segment, and then you jump off and go back here and you build a small segment. And that is what's called discontinuous synthesis, and it has a few consequences, right? So this strand that's just built is called the leading strand. So this is the leading strand. This strand that is built discontinuously is called the lagging strand. It takes a little bit longer to synthesize the lagging strand than the leading strand, so it, lags a little bit behind. Because you've got to keep doing this, jump back and then go forward and then jump back and then go forward thing. The other consequence is that here on the leading strand you have one solid, just boom, thing of DNA, strand of DNA. On the lagging strand, what you end up is a whole bunch of small fragments of DNA, and these are called Okazaki fragments. And these Okazaki fragments have to be linked together, otherwise they'd fall apart. So you need an enzyme that's going to come in and connect that, and connect that all together. And that connector enzyme is called DNAligase. To ligate something means to attach together. A ligand is something that's attached to something else. So ligase is an enzyme that attaches two things together, and DNAligase attaches two strands of DNA together. Now note up here, right? So this is our replication bubble, and we've got two forks. Let's take a look at what the lagging and leading strand is. So here, remember, we always build on to the 3' end. So if we look at this strand here, that is going to be a leading strand. because, as you see, this end that you're building onto is the 3' end. So that means that this right here is going to be a lagging strand. Now, if we take a look in the opposite direction, right, on the other fork, this over here, that's a 5' at the end right there. So this is going to be a lagging strand, and this is going to be a leading strand. So note that which strand is leading and which strand is lagging switches depending upon which replication fork you are on. So the DNAligase that I was talking about, DNAligase works by, you've got 2 strands, which you see here. Here would be 2 Okazaki fragments. Note, this is the 3' end of one, 5' to 3'. This is the 5' end of the other, 5' to 3' that direction. And what happens is the enzyme, and usually ATP, because this requires energy, is what it does is it attaches an extra phosphate onto this, and then it using the ATP, and then it uses the energy of that extra phosphate to create a bond from there, and then you'll have another phosphate here, and that phosphate will leave. And you end up with a phosphodiester linkage. That means that you have a phosphate linking to hydroxyl groups together, and all phosphate bonds in DNA are phosphodiester linkages. So, now that we know, generally speaking, the chemistry of DNA synthesis, let's look at how it's done in prokaryotes. Right. This is a pretty complicated picture, and we're going to go through it in small pieces. All right? I'm going to give you sort of an overview, and then we're going to go over each of the cast of characters. So here you see, this is the replication fork right here. This is the new strands being made. Note that this strand is being synthesized 5' to 3', so that's going to be your leading strand. This strand is being synthesized that direction, 5' to 3', so this is going to be your lagging strand up here. So, the important components of the replication complex. These are all different proteins. They're usually going to be all hooked together, but they're all technically separate proteins. Helicase. Helicase unwinds the helix. It takes energy to do it because it takes energy to unwind a helix. So it's going to sit at the front, right where the strands are being separated, and it's going to unwind the strands. Topoisomerase. Anytime you, unwind, if you've got 2 things that are sort of, wound together, and you pull them apart, what you're going to end up doing is making the winding tighter farther down. What that would end up doing is supercoiling the DNA in front of the enzyme. And eventually the DNA would get so supercoiled that you can't pull it apart anymore. Hence topoisomerase. Topoisomerase sits in front of the helicase. It moves a little bit in front of the helicase, and what it does is it will create a single-stranded break, a nick in the DNA, and allow the DNA to unwind its supercoils. So it'll go forward and unwind the supercoils in front of the replication complex, preventing the DNA from getting too supercoiled. Single-strand binding protein. Single-stranded DNA is not something you normally find inside of a cell. Generally speaking, if cells find single-stranded DNA, they assume it's from a virus and they destroy it. However, when you're making your DNA, there's going to be a short section of single-stranded DNA where, like, after it's been unwound, but before the new strand has been made. You don't want that to get destroyed, and you also don't want it to start binding to itself, which DNA is perfectly capable of doing. So there are single-strand binding proteins that basically bind to the single-stranded DNA, keep it from interacting with itself, and also keep it from being destroyed by nucleases that destroy single-stranded DNA. So, next. Primates. This is important. For whatever evolutionary reason, DNA polymerase can't start a new strand. DNA polymerase cannot begin a strand of DNA. It can only add on to the 3' end of a pre-existing strand. So, we need to make a new strand of DNA. But DNA polymerase can't do that. On the other hand, RNA polymerase can. RNA polymerases do it all the time when they transcribe DNA into RNA. So what you do is, if this is your DNA strand, right, and this is, say this is the 5' and that's the 3', and you want to add some DNA to it, what you have to do is lay down, again, 5' to 3', you have to lay down a little bit of RNA first. So you have to prime it with RNA. And then you can add DNA to the end of RNA. So you can continue it with DNA. But since DNA can't start on its own, you got to have a little bit of RNA at the beginning. That little bit of RNA at the beginning is what's called the primer. And primase is an RNA polymerase that lays down that primer. It doesn't stay in the finished product. We'll get to what happens to it later. Okay, then we got the big guy. This is the DNA polymerase core. And technically, it is the DNA polymerase 3 core. In bacteria, the DNA polymerase 3 is the main DNA polymerase. Why isn't DNA polymerase 1 the main polymerase, I hear you ask? Because it wasn't the first one discovered. They were named in the order that they were discovered, not in the order in which they're important. DNA polymerase 3 is the main polymerase. 1 and 2 exist and do other stuff. And DNA polymerase 3 actually has, like, what's called a core. All right, so imagine, like, the core is the super important region. And then it has, like, a whole bunch of other stuff kind of attached to it, hanging off. And when we talk about the core, we're just talking about this central region. When we're talking about like this, the core plus all of the other stuff, that would be called the holo enzyme. The core is made of 3 subunits: alpha, epsilon, and theta. Alpha is the one that actually catalyzes the polymerization chemistry. It's the one that actually adds a new base on. So that's the fragment that actually does the polymerization. You would think that that's the only thing that you need, and technically it is. You can isolate DNA polymerase 3 alpha all on its own, and it will polymerize things, but not very well. All right? These two are here because biology is messy, and it screws up a lot. Sometimes you put the wrong base down. Sometimes you got like A, T, C, G, and like you're laying down new bases, and you put down a T, and then you put down an A, and then you put down a G, and then you put down another G. Well, DNA polymerase 3 theta detects G to G. That's bad. We don't do that. And DNA polymerase 3 epsilon has what's called 3' to 5', right? So remember, that's your 5', that's your 3', because you always make DNA 5' to 3'. This has 3' to 5' exonuclease activity. Exo means, well, so nuclease means to remove a nucleotide. Exo means to remove a nucleotide from the end, and this means that you're going to be removing it from the 3' end. So what we're going to do is take this G right here, and we're going to remove it. Let's go ahead and remove that. It's basically a backspace button. And then we're going to put the right one down this time. So theta is going to detect incorrect base pairs. Epsilon is your backspace. Alpha is your main polymerase. Now, remember how we said that there is a DNA polymerase 1, and what it actually does, it's not part of the core enzyme. Let's say we've got our old DNA. I'm going to do old DNA, new DNA in black, and new DNA in blue. So let's say this is 5, 3, 5, 3. All right. So our new DNA, 5, we're going to need a little bit of an RNA primer. RNA is in red. But then this is going to be our leading strand. And so it's just going to go 5' to 3'. Now this other one, this is the 3', 5' is going to be over here. This is our lagging strand. So we're going to need a little primer, DNA, primer, DNA, primer, DNA, and like that. But you can't just leave all of that RNA in there. So what DNA polymerase 1 does is it sits on this site, And it's going to take a small bit. Let's actually zoom in here. And what it's going to do is take a small, like one base of RNA off, and then lay down a base of DNA. And then it's going to take another base of RNA off, and then lay down a little bit more DNA. And it's going to do this one base at a time until all of the RNA has been removed and replaced with DNA that's then going to be ligated together. So this DNA polymerase 1 needs two functions. It needs a 5' to 3' exonuclease activity, because what that's going to do is that's going to remove the base in front of it, the RNA base in front of it. This is similar to like the overwrite feature on a keyboard, so that you lay down a new base in place of the one in front of you. It also has a 3' to 5' exonuclease activity because it still makes mistakes. So it still has that backspace. But what it's going to do is pull up the RNA base in front of it, clip that off, lay down a DNA base. Pull up the next RNA base, clip that off, lay down a DNA base. And it does that replacing the RNA primers with DNA. And so where would you find a lot of DNA polymerase 1 associated with? The lagging strand or the leading strand? What do you think? So DNAligase, we already basically talked about, but this is another one of our cast of characters. It's going to be necessary to, after DNA polymerase 1 has replaced the primers with DNA, you're going to need DNAligase to attach one Okazaki fragment to the next Okazaki fragment. And now we have the hollow enzyme, right? So, first off, remember we had the core enzyme, which is alpha, epsilon, and theta. This is the core enzyme. So is this. Note that there's two of them attached together. That's because the bit that's synthesizing the leading strand and the bit that's synthesizing the lagging strand are attached together. They're in the same holoenzyme complex. And they're moving the same directions. What you see down here, this is tau. Tau's a linker. It's what's going to connect the two core enzymes together. And then you have the beta clamps. The beta clamps are like hands that clamp around the strands. And when they clamp around the strands, they keep the enzyme from letting go. This one is going to be around the leading strand. The lagging strand is going to be this one. And you know how I can tell that? Because of these, the delta and gammas. Once the leading strand clamp is clamped on, it just stays on and keeps making leading strand. But remember that the lagging strand clamp is going to have to let go and then reattach at the beginning of the next Okazaki fragment. These here basically help mediate that clamp letting go, moving to a new place, and then clamping back on. And it's going to have to do that many times as it synthesizes the new strand of DNA. So here you can see everything put together. All right? Helicase is unwinding the DNA. In front of it, DNAligase, which is a topoisomerase, is relieving the supercoils. Here we have a few single-stranded binding proteins, and this is our core polymerase, and it's going through the core polymerase to the clamp. And as you move, remember, we're moving this direction. As you move this direction, you add on boom, 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 boom. New bases. That's the leading strand. Now you have the lagging strand. And it's a little bit weird because you have the same enzyme in the same orientation, which means it's going to have to be making DNA the same direction. But remember, it has to make it the opposite direction. So how does it do that? It kind of pulls out this big loop and basically throws the loop in front of it. Right? So if this is the green is going 5' to 3' this way, this green is going 5' to 3' this way. So you're synthesizing new DNA this direction, and you're going to want to be synthesizing new DNA that direction on this other strand. So you have to loop this big bit out and around so that you've basically got it twisted and facing the right direction. And you have your clamp, and you have your enzyme, and now you're making a new Okazaki fragment right here. You see the primase associated with the lagging strand, which is going to lay down a new RNA primer. At the beginning. And then the primase disengages, and you make more DNA until you run into the next Okazaki fragment. And then this clamp will let go, and you've got this back here, which is basically a part of the mechanism that attaches a new clamp. It's going to attach a new clamp here, and then put that new clamp on, and then it's going to wheel out a new strand, and then from this new clamp point, so this is going to be transferred to here, and from this new clamp point, you're going to start making RNA from the primase, and then DNA until you run into the next Okazaki fragment. All right. That's the mechanism for how the DNA synthesis actually happens. But how does it get started? Remember, we said that it happens at the origin of replication, which has these repeat regions that are usually going to be AT-rich, and they're near these 13 base repeats. They have a specific sequence. What happens is these AT-rich regions here they bind a special protein called DNA-A. And so DNA-A starts to build up along those. And DNA-A will bind to this AT-rich sequence, and it will also bind to more DNA-A. So what you end up with is a whole bunch of DNA-A binding to each other and binding to these 13 base pair repeat sequences further down, and they're going to bend the DNA back over on itself. Once you've bent the DNA back over on itself, that puts strain on the DNA, and that causes the DNA to be more likely to breathe, to split open. So you get strand separation that begins at the 13 base pair repeats as they open up due to the strain, and once they've opened up, your helicase can jump in. And once the helicase is in there, it will keep the DNA strand open and will begin unwinding it in both directions. So this is called the closed complex, and this is called the open complex. And this whole thing here is what's called the initiation complex, or the replication initiation complex. There's one other type of DNA replication that is done in prokaryotes. Some prokaryotes, most, have small circular DNAs called plasmids. They're much, much smaller than your normal chromosome. And they're replicated in a different way. They're replicated through what's called rolling circle replication. With a normal replication, you just pry the two strands apart. You never actually nick or cut the DNA. But with rolling circle replication, you do. You put a nick in, a single-stranded break, and then one of the strands will start rolling away. So here you can see this strand has started rolling away, and this plasmid has started circling. Now, what you have is a DNA replication enzyme here and a DNA replication enzyme here. They're actually linked together. And they lay down, as this rolls, DNA 5' to 3' there, and it actually attaches here, and it'll lay down 5' to 3' there, so that the new strand that's emerging is going to be double-stranded, and the template strand, the one that wasn't cut, as it rolls around, it gets a totally new strand of DNA attached to it, and then you cut this and form it into a circle. There's actually two ways that this can be done. It can be done so that you synthesize both of the new strands at the same time, like I've shown here. Or it can be done so that you just synthesize the new strand on the template strand, and what you're left with is single-stranded DNA that then goes somewhere else, like perhaps to another bacteria, forms itself into a single-stranded circle, and then an enzyme in the new bacteria is going to add the second strand to it.